Hey everybody, it's Mrs. Mancuso, Merm here. Today's video for AP Bio is about genetic technology. So this is a huge area. I'm going to give you guys a chance to do a bunch of research to learn more about fields that you're interested in, just because it's so gigantic. So the basic idea is that genetic technology is all about analyzing, so like figuring out the sequence of genes and figuring out what genes are present within certain chromosomes. And then the forms of the gene, the alleles, what are they and what do they code for? So then using that information, analyzing what you learn about the DNA to manipulate it. So a really basic idea is to find and isolate genes and then reinsert them, put them in another organism or put a corrected form back inside of an organism. All right, so many areas to explore. We're going to focus this video on a few of them, and then I'm going to give you class time so that you can figure out what you're most interested in and what you want to learn about. So the ones that I'm going to do today: recombinant DNA, transformation, cloning, electrophoresis, and CRISPR. I'm going to stick with those 5. So the first one, recombinant DNA. It's exactly what it sounds like, getting a new combination of DNA. You want to clone a gene and put it into a plasmid, and I'm going to explain what a plasmid is in a second, and then you want to be able to take that plasmid and put it into another organism. So goal, DNA from another source incorporated into a cell, and then it has to express itself too. You can't just put it in there, it has to be turned on. And sometimes you need a clever way to get it to turn on, or a certain presence of a substrate. All right, examples. So we now have crops that are drought resistant and insect resistant and fungal resistant. And that's because genes from other organisms that are drought resistant naturally and that are insect resistant naturally and are fungal resistant naturally, they isolated the genes and put them into a plasmid and inserted them into like corn and wheat and things like that, so that the crops aren't destroyed. And then another really common example, which used to be done so much for diabetic patients, is that they took the human insulin gene and put it into bacteria so that bacteria would make insulin that pharmaceutical companies could mass produce and sell. Now they do it more synthetically, but that was a great example for a while. All right, next one. The thing about recombinant DNA is you have to have a plasmid as a vector. A vector is a way to transport and bring something into a cell. So plasmids are perfect because those are circular pieces of DNA. They're the extra DNA that you find in a bacteria. So it's going to use a plasmid when you do recombinant DNA. You have to have restriction enzymes. So restriction enzymes are specific enzymes. There's a whole bunch of them, like Echo R1 and BAM and Hinge3. They all have these different names. They each cut DNA at palindromes. Palindromes is a repeating pattern that's read the same in both directions. And it cuts at those specific sequences to give you sticky ends. So let me give you an example here. If this is my DNA, these are the hydrogen bonds between the bases like this. If I cut it with a restriction enzyme, it would look like this. This is a sticky end, so that part is open. And then the other piece is going to look like this. This is open. Now, those sticky ends can then insert an extra gene in the middle of there and insert that into your plasmid. All right, so both the donor and the plasmid have to be cut with the same restriction enzymes. If they're not cut with the same restriction enzymes, they're not going to have the same sticky ends, so they're not going to connect. So for example, if this is like ATCGC, then over here is going to be those complementary letters. And then when I have a new piece, it's got to be cut with the exact same one so that their letters match. And then the thing that connects them is ligase, which I told you when we learned about ligase back in DNA replication, it was going to come up again. Here it is coming up again. So ligase is our DNA glue. It connects the DNA pieces from the new gene to the plasmid. And when it does that, it's really connecting the sugar phosphates, and the sugar phosphate bonds are called phosphodiester bonds. All right. Now that you have a plasmid with an inserted piece of DNA, the host can now receive the recombinant plasmid. And then really important, it's got to be able to transcribe it and translate it so that it makes the new protein from the original donor. And sometimes that requires an activator to turn on transcription of the gene. So let's just draw it out. So we're going to say that this is my plasmid donor. And I'm going to make my plasmid donor have blue plasmids. Okay. I'm going to treat it with a restriction enzyme. So now it's like this. It's open. It has those sticky open spots. And then over here, this is my DNA donor. So this has the gene that I want to insert. So maybe it's a gene so that it'll glow in the dark, or it's a gene for resistance, or a gene for, you could pretty much come up with anything. So that DNA donor, probably it's going to be something linear, because it's probably not going to be a prokaryote. So there's the little piece. And again, it's got the sticky ends, meaning one part of it has that little opening. Okay. When you put these two together, you end up getting a plasmid that has the inserted gene, like that. And actually, if I drew this even better, you'd be able to see that the red is like this. So that's where the sticky end connects. And then the blue is like this. So there's the other sticky end connecting, and there's the original. So this is my recombinant DNA plasmid. Recombinant DNA plasmid. So again, this plasmid is just so that I can insert it into something else. So now I can take this, put this whole thing into another organism like a plant, and then that plant is going to be in, let's say it's insect resistance, the red gene that I put in it. It will now take up those that DNA into its cells, and when it transcribes this DNA into RNA and then translates it into proteins, it's going to have the insect resistance in the protein. Here's a little example, like one of their drawings. So there is my bacteria that I'm getting the plasmid from. I cut the plasmid open and I insert the gene. They both had the same sticky end. So now I put this blue gene in the middle here. I ligase it together. So that's my plasmid. And then I get the new gene into a cell. So basically you can do this in vivo or ex vivo, meaning you could insert it into a cell outside the body. That would be ex vivo. Or you could actually inject the plasmid into the body, and then it will take it up into the cells within the body, and that would be in vivo. All right, transformation. This is probably my favorite lab of the entire year. Not probably, it 100% is my favorite lab of the year in AP Bio. So transformation is a process that is specific to bacterial cells. And so I always call them like, they're little kleptos. Bacterial cells will take in all of the stuff from their surrounding environment. So when they take it in, they can take in plasmids, and plasmids can be formed the way that we just talked about. So heat shocking bacteria, meaning cold, warm, cold. That change of temperature, or warm, cold, warm, that change of temperature changes the permeability of the membrane, makes it more likely for them to transform, meaning take in something from their environment. When you expose heat shock bacteria to desired genes in plasmids, some of the bacteria are going to take in the plasmids and express those genes. Bacteria is never 100% transformable, meaning not everyone will take in the plasmids from the environment, but many of them will. You can actually figure out how the percentage that were transformed just by seeing which percentage got the gene that you were exposing them to. So what I think is super interesting and relevant is that we know antibiotic resistance spreads so quickly amongst bacteria. So like we always used amoxicillin, and now many bacteria are amoxicillin resistant, which means that if you take it, it's not killing the bacteria. So then you go to Cefsil or some other antibiotic, And bacteria are becoming more and more resistant because they pick it up from their environment. So let me just make sure you understand what I'm saying. Here's bacteria. Here's the bacteria's normal circular DNA. And then it has these extra little circles. Those are plasmids. And out in the environment, there are plasmids from other bacteria. So this bacteria could let out its plasmids, and it also could take in other plasmids from the environment. So the movement of plasmids in and out of a bacterial cell is called transformation. And then we're going to do this in our lab. So that's going to be super fun. All right. I was going to draw a picture of how we're doing it in the lab, but this video is going to be super long, so I'm just going to move on to the next one because we'll do more with the lab. All right, cloning. So cloning is when you make a copy of something. You need to have a host, which is going to be the surrogate mother, and then you have to have a donor. So I'm specifically talking about cloning organisms right now, not just cloning genes like we were doing with the plasmids, but getting a whole new organisms like Dolly. I'm sure you've heard about Dolly the sheep. So in order to do that, you have to remove the diploid nucleus from a somatic cell. So a somatic cell is a body cell. So just a normal diploid nucleus. Take it out of the donor organism. Then you got to take an egg cell from the surrogate mother, so the one that's going to end up pregnant. Take one of her egg cells, take the haploid nucleus out of it, and replace it with the diploid nucleus from the donor. So you're literally switching the egg for a diploid nucleus. Reinsert the now diploid cell into the surrogate mother. And then if the zygote develops, so it doesn't happen 100% of the time, it's not 100% effective. Otherwise, everyone that tried having in vitro fertilization, it would work, and it doesn't work with everybody. So take that zygote, and if the baby develops, the baby is going to be born identical to the donor because it has the exact same DNA. All right, next one, electrophoresis. We also will do a lab on this. So this is separating pieces of, you can do it with DNA, RNA, or proteins. It's based on the size of whatever molecule you're doing and the charge. And it's used in crime scenes. So when they take the suspect, or multiple suspects' DNA and compare it to DNA that's found at the crime scene to see which one is a match. It's done in paternity tests. It's done in detecting genetic disorders. So let's say your mom has breast cancer and you're like, I want to know if I have the BRCA gene. You could have a genetic test done to see if you have the same gene. It's also for identifying evolutionary relationships amongst organisms. Like there are certain animals and they're like, well, is that more related to a horse or a zebra, or what is it related to? And they can study the DNA and see what it's related to. All right, so in order to do it, it requires DNA to first be cut with restriction enzymes. So again, those are the ones that cut at the very specific palindromes and give you the sticky ends. Then you place those fragments into DNA, an electrophoresis gel. So the gel is an agarose gel, which means it has little spaces for molecules to move through. And then you cover it, turn on the electricity, and the DNA is going to move from the negative electrode to the positive electrode. And again, you'll see that happening. You can literally see the bubbles and see the movement. You put a dye in there so you can trace it and follow it. And then each resulting band is a piece of DNA. The further it travels, the smaller the piece was. When you see really thick bands, that just means you had a lot of fragments that were that size. And you can determine the size. So if you want to know, like, how large was this fragment, you can run a standard on the same gel. You know the number of fragments, you know the sizes for the standard, and if your sample moves the same distance as a fragment from the standard, you know it's about the same size. So what you do is you use logarithmic graph paper, you plot your standard, and then you use it to determine the relative size of anything else that was run on the same gel. So I'll show you what I mean by all of this. So first of all, here is my gel. These are the wells. This would be the negative end, and this would be the positive end. You load your DNA that's already been cut with restriction enzymes into each of these. So I'm going to run a standard here, and I'll run, let's say I run the crime scene here, and I run a suspect. I better run another. Suspect 1 and suspect 2. So I only run the standard because I want to know the size of them. So let's say I know the standard gives me 4 pieces. 1, 2, 3, 4. And then crime scene. Let's say this one gives me 3 pieces. So from that same piece of DNA, or from a piece of DNA that was cut with the same restriction enzymes, I get 3. And let's say suspect 1 gives me the exact same bands. That's a match. That's really what you're looking for. And let's say suspect 2, those bands are different, or maybe it cuts it in more places. So you know it's not suspect 2 because the bands don't match up with the crime scene. All right, and then over here, I want to show you how you use the standard. So you take your graph paper, and over here are the number of base pairs. And over here is the millimeters traveled. So how many millimeters does it travel from here to here? So you plot your standard. So you plot your standard and you get a line like this. Then I know for any distance. So like, let's say this one right here, it doesn't match up with one of these lines, but I could measure the distance, find it here, go up, and this is going to tell me about the number of base pairs that are in that piece. All right, my last one for this video is CRISPR. CRISPR is new technology in that they really started using it in probably around 2015, but now in 2020, it is now just becoming more of a conversation piece and much more of super interest to people for what it can do. So it's for gene therapy and for gene editing. So let's say you inherited a genetic disorder, like cystic fibrosis or something. This could potentially be a way of cutting out the cystic fibrosis gene and replacing it with a good gene. So really just cool technology, cool stuff. Now, how they found it is wild. It is actually part of the immune system of a bacterial cell. So bacteria actually have this CRISPR, which I forgot exactly all what the CRISPR stands for. You'd have to look it up. But it's basically this section of DNA that codes for Cas9, which is an enzyme, and its entire job is to cut viral DNA out of a cell. So bacterial cells can easily be infected by bacteriophages. So if the bacteria can recognize, like, ooh, this little segment here, that came from a virus, let's get it out of here. So that's what CRISPR does. So CRISPR is small nucleotide segments that code for Cas9, and Cas9 cuts out viral DNA. So if you could use this cutting technology in your cells, you could cut out genes and insert correct ones. So it's really about cutting. It's really about cutting genes at specific spots, taking out what you don't want and putting in what you do want. So it's using technology that was found in a bacteria and using it in other organisms to replace genes. All right, that is the end of our technology video. See you guys soon.